So right now in this slide, you can see, so the population of India. Okay. So in 2018, it was 1.34 billion. And we are expecting in 2050, so it will cross 1.70 billion. So if you see the food requirement in by 2050, so it will be 333 metric ton. And if you see the productivity, if you see the production plateau, so yield plateau is almost reached. That means it is stagnant now. So the productivity is right now, it's not increasing as the population is increasing in the geometric progression. So in 2015, what alarming? So will present agriculture practices be enough to meet these challenges of future needs? Is it not viable to have the preparedness for new technologies which can produce more from less? Because we have to think for such a newer technology, we have to adopt such a technology which can satisfy our future needs of population. So can new technologies like biotechnology able to meet the expectations? So each green revolution is backed by technology. So till now, each agriculture revolution. So the green revolution, the blue revolution, the white revolution. So everywhere it is backed by some technology. Hence, let us have faith in technology since the reliability of future. So thus the country needs adoption and infusion of biotech crops, that is GM technologies, including the newer ones of gene editing and so on. So one of the economists, Jeffrey Sachs, so in 2000, he said that it is not only the ideology, but the technology which the system needs. Okay, so every revolution, it is backed by the technology. So till now, you can see the various technologies which brought the revolution in India. So first it was the mechanization. So there was the uses of the tractors. Then thereafter there were the fertilizers. So the Fritz Haber in 1913, who invented the ammonia, okay, synthesized ammonia, and that can be used as a nitrogen fertilizers. Okay, that can be used as a nitrogen fertilizer. So there was a so this technology has been implemented by the Fritz Haber. Then there was an era of pesticides, that's chemicals derived from hydrocarbons, enable the farmers to grow high density crops year after year without severe loss to the insect and pest. Then there was an era of seeds, like in 1960s, dwarf wheat variety rapidly replaced with the South Asian subcontinent hunger, with surplus in the green revolution. Okay, so these four technology which has brought a revolution in India. So we should not forget about these two legends. So M.S. Swaminathan, who is well known for the green revolution in India. Then Norman Borlaug. So we should not forget these two scientists when we talk about the agriculture. So what are the various technologies and how revolution happens? So in in 19th century there was a traditional farming. So then mechanization came, various tractors came, various implements mechanization in agriculture came. Then then in 1970s, so dwarf varieties has been implemented in the agriculture, and that was the era of green revolution. And in 20th century we can see to the biotechnology, and we can have a lot of hopes from that genetic engineering and biotechnology in the field of agriculture. So here we can see, so there were various revolution in the Indian subcontinent. So food grain revolution, we call it as a green revolution. Then fruit and vegetable production, we call it as a golden revolution. Then fish production, we call it as a blue revolution. So there was an increase in the egg and poultry production, we call it as a silver revolution. There was increase in the milk production, we call it as a white revolution. So green revolution, so the high yielding semi-dwarf open pollinated varieties, which played a major role in the green revolution. Then there was an era of hybridization. So genetic male sterility, cytoplasmic male sterility. So that has helped to create various hybrids in various crops. Then there is an era right now. There is an era of biotechnology. So with the help of biotechnology and genetic engineering tools, so various plant cultivars has been developed for various traits like insect resistance, herbicide resistance, disease resistance in various crops. And now, as there are various pros and cons to the field of genetic engineered crops. So right now there is a new breeding technology which is coming up, that is a CRISPR-Cas. Okay, so that is a genome editing technology. Okay, genome editing technology. So there is some advantages over the GM crops in this genome editing technology, that is a CRISPR-Cas. So we have to always think for the newer technology. We have to implement the various newer technology in the field of agriculture if we have to fulfill the demand of Indian population by 2015. So today I have seen in you can see here the today's agro one. Okay, so in today's agro one, so you can see so it is 17th February 2022 paper. That means today's paper. So I have seen that various farmers actually they are now they are trying to make an movement so that the GM crops can be in the food channel. So they are trying to pressurize the government for incorporation of the GM plants in the field of agriculture. So that's the importance of technology, okay, which are actually the helping for the various farmer. So genetic engineering. So this is the major innovation in the crop biotechnology, which provides exclusive solution for various viral diseases, cryptic insects, salinity stress, then drought stress. Then to mitigate the various climate change, we can develop the variety to mitigate various climate change with the help of biotechnology or genetic engineering. So in general, if you see the definition of genetic engineering, okay, so genetic engineering is one of the tool of biotechnology. Okay, there are various tools of biotechnology, but genetic engineering is one of them. And with this, we can producing the various transgenic plants. Now, if we see the transgenic plants, actually its name said that it is a transgene. Transgene means what? So the gene which has been incorporated into a cultivar from the other species. Okay, the gene which has been incorporated into a species into an organism from other species. So we call it as a transgenic. If the gene source of gene is others, if we will take the gene from other species and if we will incorporate into a particular organism, so it is a transgenic. Okay, and if we take a gene from same species and we are incorporating the gene in the other cultivar of the same species, then we call it as a cisgenic. Okay, so you should remember the difference between transgenetic and cisgenetic. So transgenetic means we are taking a gene from other species and we are incorporating into a particular organism. Then cisgenetic, so we are isolating a gene from same species and we are incorporating that gene into a same species. So it is a cisgenetic. Okay, so here we can see gene from foreign sources. So you know the very good example, that is Bt cotton. So where that Bt gene has been incorporated from the bacterium, okay, that is a soil bacterium, that is Bacillus thuringiensis. Okay, so it's a source is different. That's why we call it as a transgenic. Okay, and if I isolated the gene from rice and we are incorporating that gene into a rice by using genetic engineering tools, then we call that plant as a cisgenic. Okay, we are not incorporating the foreign gene there. So you know a lot about the Bt cotton. So the gene has been isolated from that Bacillus thuringiensis and it is incorporated into a plant. And when that bacterial gene is expressing, that means bacterial gene expressing means what? So gene is expressing means what? So gene is expressing means that gene is producing mRNA and that mRNA is getting translated into a protein. Okay, so here that Bt gene get expressed by the process of transcription and translation and that gene will produce a protein. So which is that protein? It is a Bt protein, and that Bt protein actually it is a toxic to the various insects. So what happens when insect feed on that leaves or that plant? So that along with that leaf, that protein is also getting ingested by that larvae or insect, and once that protein is getting ingested into the stomach or into the midgut of an insect, or into the gut of an insect, then that will become active in the alkaline pH. Okay, so it needs alkaline pH for its activity. Okay, so the Bt protein, it is almost active in the alkaline pH. So in this way, that insect will get died due to this toxic Bt protein. Okay, so this is a general overview of that Bt cotton plant. Now, what is actually the GM crop? So we talk about genetically modified crop, transgenic crop, but what it is? So it is a crop which has a gene artificially inserted into it from another species. So that may be even unrelated, okay? And it gives similar properties of that particular plant, despite of incorporating a foreign gene into that particular plant. So in this way, we can call that plant as a GM crop or GM plant or genetically modified plant. So the question comes, are there other GM crops in India? So answer is no, other than Bt cotton. So right now government has not given the approval for other GM, for cultivation of the other GM crops in India. Okay, indeed India gets its first GM crop. So the first GM crop variety approved for commercialization was Bt cotton. So the trade name of that Bt cotton was Bolgard 1. Okay, the trade name of that Bt cotton was Bolgard 1, and which provided the immunity against the pink bollworm developed by Monsanto in 2002. So that Bolgard 1 has been approved by the government of India for commercialization in the year 2002. So the first GM crop in India has been incorporated for the cultivation in the year 2002. So thereafter in 2006, okay, thereafter in 2006, so Monsanto released Bolgard 2. Okay, so Monsanto released Bolgard 2 in the year 2006, and right now you can see the India has become the world's largest producer of cotton. Okay, right now we can see the India is the largest producer and exporter of the cotton. So what is the benefit of that GM crops? So if we see by the economic view and environmental view, so increased crop production. Okay, so the GM crops has been increased the crop production, and it's valued by 224 billion US dollar, and it providing, so if you talk about the environment, it providing a better environment by saving 776 million kg of pesticides. It's also helped to reduce the CO2 emissions. It also helps to conserve the biodiversity by saving 231 million hectares of land. Okay, and it also helped to increase the profit of various small scale farmers. So if we talk about the genetically modified crops, so when we talk about the genetically modified crops, so which are the, if you talk about the genetically modified crops all over the world, so which are the countries which have the largest production of these transgenic genetically modified plants? So if you see here, so US come first, so where the genetically modified crops are grown in the largest area. Then it is followed by the Brazil. Then it is Brazil is followed by the Argentina, and then at fourth India come. Okay, so these are the 5 topmost countries where you can see the genetically modified crops growing on the largest area. So the first is United States, then it is followed by Brazil, then Brazil is followed by Argentina, and India comes fourth. So the US is the first in the cultivation, the US is the first who cultivates the genetically modified crops on the large scale. So we should remember that order: United States, Brazil, Argentina, and India. So you can see the area of GM crops in 2019, which was 190 million hectares. Okay, and 43 countries imported foods and products derived from that GM crops. So what are the major traits of that GM crops? That are insect pest resistance, herbicide tolerance, virus resistance, male sterility, and modified oil quality. Means India is also importing the various product of edible for the edible purpose from various GM crops of other countries. Okay, even though we are cultivating only the BT cotton, okay, for insect resistance, but we are importing the various byproducts of that GM crops from other countries. So if we see the historical background of that genetically modified crops, so the first GM crop was produced in 1983. So which was the transgenic tobacco. Okay, transgenic tobacco. It was only the that means transgenic tobacco has been produced in 1983. Okay, the first GM plant, but actually the commercialized transgenic plant, it was the tomato. Okay, the first transgenic plant which has been commercialized, it was the tomato. So in 1994, the US Department of US Food and Drug Administration approved the Flavor Saver tomato for commercial release. So that Flavor Saver is the trade name for that tomato. Okay, right now we can see the BT cotton. So the trade name, it is being sold by the trade name of Bolgard 1, Bolgard 2. Okay, so similarly, for this tomato, the trade name was Flavor Saver. So for what purpose that tomato has been developed? So it is developed to enhance the shelf life. Okay, to enhance the shelf life of a tomato. You know, the tomato, they are the, that means perishable vegetable. So immediately it will that means due to the high rate of respiration, so it will be immediately, it will get spoiled. So how to enhance the shelf life of a tomato? So by using this genetic engineering, so in 1994, US developing United States has developed a tomato which has the longer shelf life, and its trade name was Flavor Saver. Then in 1995, the BT potato has come. Okay, and BT cotton has been also approved. Okay, it was only approved so for commercial cultivation in US. Then in 1996, first GM maize producing a BT cry protein was approved. Okay, so in 1996, the first GM maize producing a BT cry protein was approved. Okay, then in 2000, the first two GM rice varieties, both herbicide resistance, called LL rice 60 and LL rice 62, were approved in the United States. Then in 2000, vitamin A enriched golden rice was introduced in India. BT cotton was released in 2002. So what we have to remember here? So actually, the first transgenic plant which has been developed, it is a tobacco, but it is developed only for the research purpose. Okay, at that time, but actually the commercialized plant first GM crop has been come in 1994 in the United States, and that is for the enhanced shelf life, and that trade name was Flavor Saver tomato. So the first GM food, that is I told you, the first commercially grown genetically modified food crop, it was Flavor Saver tomato. Okay, and what is that means what is the purpose, what is the character? So that means delayed ripening, we call it as a delayed ripening or enhanced shelf life. Okay, it has been developed by antisense RNA technology. Okay, gene silencing technology. So which is one of the method of that genetically engineered to make the genetically engineered plants. Okay, so the Flavor Saver tomato, it's developed by the antisense technology, antisense RNA technology, which is the method of gene silencing, and where we can see the enhanced shelf life of a tomato. Okay, where we can see the enhanced shelf life of a tomato, or we can see the delayed ripening of tomato. So in 1994, it has been approved by US Food and Drug Administration Department, and it has come in the market by the company Calgene. Okay, so Calgene, we have introduced it into the market. So you should also remember the name of that firm. So Calgene company, the company which has introduced that particular tomato, transgenic GM tomato in the US. So like tomato, we also should remember what is the first genetically modified ornamental plant. So that is carnation. So whenever we talk about the flower plant, ornamental plant, so it is a carnation, which is the first genetically modified plant. And what has been modified in the carnation? So it is a color of that flower. Okay. So what we have modified in this plant? It is a flower color. Then if we talk about the trees, okay. So trees, the popular were the first genetically modified trees. Okay. So the GM, that means popular transgenic poplars has been produced with the BT genes. Okay. So which is the first tree where the transgenic plants has been made, or so it is a poplar, and for which gene? It is a BT gene. Then the first transgenic cereal crop. So which was the first transgenic cereal crop? It was the BT maize. So we are talking about all over the world. Okay, so the first transgenic cereal crop released commercially was BT maize, and the first tree, transgenic tree, or first GM genetically modified tree, it is developed. The name it is developed in poplar. Then first genetically modified ornamental plant, or which has been developed in the carnation, and it is developed for the flower color. So we can see here. So as I told you, the US is the top leader. That means United States of America. That means who are cultivating these transgenic plants or genetically modified plants on the largest scale. Okay. So right now, if we see at the global level, so GM genetically modified technology has been used in the various non-food crops like cotton, poplar, alfalfa, eucalyptus, tobacco, carnation, rose, petunia, and also in case of food crops like maize, soybean, potato, tomato, papaya. That means for various traits. So here we can see. So right now, if we talk about the world, so 191.7 million hectares is under the biotech crops. Okay, that means near about 200 million hectares of area is under the biotech crop. Now, if we look at here, so what are the major biotech crops? We have seen about the US, we have seen about the Argentina, Brazil, and India, which are the topmost which are the topmost countries who are cultivating the genetically modified crop or transgenic crops. But if you talk about the crops, which are the major biotech crops? So here we can see the soybean is the major biotech crop. Okay, soybean, and that soybean also is for the herbicide trait, herbicide resistant trait, herbicide tolerant trait. Means if you spray the herbicide, broad spectrum herbicides on that soybean, then also it will survive. Okay, so it can be useful for the weed control and saving the labor cost. Okay, so soybean, which occupies the largest area of that among the transgenic plant all over the world. Okay, so you can see here. Then it comes to the maize. Okay, then it comes to the maize, second most largest crop for the second most largest crop, GM crop under the cultivation all over the world. So maize, soybean, then maize, and then comes to the cotton. Okay, then it comes to the cotton, and then it comes to the canola, and then rest of the crops are there. Okay, so first is soybean, then maize, then cotton, and then canola, and then rest of the crops are there. So here we can see, so as I told you, the US, so which is the largest cultivar of that GM crops. Okay, so 95.9 million hectares of total area. Okay, so it's under the soybean, biotech soybean. So soybean, that is GM soybean, and for which trait? It is for herbicide tolerant trait. Then biotech maize, that is GM maize. So it is the area is around 58.9 million hectares all over the world. Then BT cotton or biotech cotton or genetically modified cotton. So 25 million hectares of area is there all over the world. Then GM canola, so 10.1 million hectares of total area. Okay. Then biotech alfalfa, so 1.3 million hectares of total area. So we can see the soybean. So it is approved in around 31 countries. Okay. Then biotech maize, it is approved in 35 countries. Then the biotech cotton, that is BT cotton, it is approved in 27 countries. BT canola, so that is approved in 15 countries. Okay. So this is the statistic. Okay. This is the statistic of that GM crops which are being cultivated on the largest scale worldwide. So which countries grows this genetically modified crop or GM crops? So it is in North America, so then Latin America, various European countries, then Asian countries like Bangladesh, Myanmar, China, Philippines, India, Pakistan, Vietnam, then Africa. So as I told you, if you see the trait, okay, we have seen the crop wise, that means soybean is the largest largest GM crop cultivated worldwide, then maize, then cotton. So now if you talk about the trait, so which is the major trait of GM crop? So it is herbicide tolerant, means with the help of genetically modified plants, genetic engineering. So herbicide tolerant trait. So the herbicide tolerant traits, the crops which are having the herbicide tolerant traits are growing at the largest scale. If you talk about the trait, okay, then it is followed by the insect resistance. Then it is followed by the other disease resistance and other parameters like improving the quality, like oil quality. Okay, so the herbicide tolerance or herbicide resistance is the major traits. Okay, focus for the GM crops. So herbicide tolerant or herbicide resistance, we talk about the herbicide tolerant GM crops or herbicide resistant GM crops. So how they have been made? Okay, when we talk about these herbicide tolerant GM crops or herbicide resistance genetically engineered plants, so how they have been developed? So here we can see the Roundup is one of the herbicide. Okay, so whenever we spray the glyphosate, okay, so whenever we spray the glyphosate, so it is inhibiting the plant EPSP synthase gene. Okay, so it's act on the glyphosate, it will act on glyphosate, you know, it is a broad spectrum herbicide, and when we spray that plant, when we spray any plant with this glyphosate, then it acts on the plant EPSP synthase gene. Okay, so which is synthase gene. So it is a gene which is involved in the shikimate pathway or amino acid synthesis pathway. So it blocks its activity, that is plant EPSP synthase gene. Okay, and then there is an inhibition of amino acid biosynthetic pathway, and ultimately if there are no amino acids are getting synthesized, so that plants get died. Okay, so what happens in herbicide resistance plant or herbicide tolerant plant? So here we can see when we spray the glyphosate, so usually it will inhibit the EPSP synthase gene of the plant. Okay, so what actually in herbicide tolerant herbicide resistance plant have been developed? So that EPSP gene has been replaced with this bacterial EPSP synthase gene. Okay, so plant has been transformed with bacterial EPSP synthase gene. So that gene actually that means it is the glyphosate is not act on that gene, okay, due to some certain mutations, and that's why the Roundup has no effect on the plant, or that herbicide glyphosate will not have any effect on the plant. And in this way, that shikimate pathway will produce the various amino acids required for the plant metabolism. So Roundup ready, okay, so the Roundup means herbicide tolerant plant, okay, so glyphosate tolerant plant, okay. So what happened here, bacterial EPSP synthase gene has been incorporated from the bacterial source, and in this way herbicide resistance plant have been developed. So here we can see the difference. So before, so there we can see the in between the crop there is a weed, and whenever we spray some Roundup ready, so that crop has been survived due to this herbicide tolerant gene, that is bacterial EPSP gene, okay. And the in this way we can control the weed easily. So if you consider the Asian countries, okay, if you consider the Asian countries, so Bangladesh is the only country in Asia which plants the biotech food crop. Okay, food crop. I'm talking about the food crops. So 5 years of cultivating BT brinjal at 2000 or near about 2,795 hectares by 30,000 farmers. Okay, so Bangladesh is the only country in Asia which are planting the biotech food crop, so including BT brinjal. So right now 3 BT brinjal varieties are released for commercialization in the Bangladesh, and 2 varieties are under the field trials. Even they are also trying for the other GM crops like lead blight resistance potato, BT cotton, and golden rice. Now all over the world, if you see the various trade name for that GM crops, okay, if you all over the world. So in case of BT cotton, you know, it is a Bolgard 1 and Bolgard 2, okay. So these are the some trade name for that commercially released GM crop or genetically modified crop or genetically engineered plant, okay. So what are they? So Liberty Lean, okay, the company or the firm which is involved in for that, it is a buyer, okay. So they have developed the herbicide resistant plant, okay, Liberty Lean. Bolgard, it has been developed by Monsanto for the insect resistance. So the cry genes of Bacillus thuringiensis has been incorporated into this cotton, okay. Then Roundup Ready. So Roundup Ready, it has also been produced by the Monsanto company, okay, for glyphosate resistance plant, that is herbicide resistant plant or herbicide tolerant plant, okay. Yield Guard. So Yield Guard is the trade name for the maize, okay. So Yield Guard has been developed to for against the European corn borer, okay. Yield Guard has been developed against the European corn borer by the company Monsanto. So you should remember the various trade name of these GM crops. So Lorical, okay. So Lorical, it is developed by Calgene in the rapeseed, okay. So Lorical also that is also you should remember. Then coming to the Bolgard 1 and Bolgard 2, okay. So Bolgard 1, you know, in Bolgard 1 only Cry1 AC gene was there, which has been isolated from Bacillus thuringiensis, and it has been incorporated in the cotton. So Bolgard 2, it's have the 2 genes, that is Cry1 AC and Cry2 AB, which provides protection against bollworm and Pseudoptera caterpillar, okay. So initially Cry1 AC is imparting the resistance to only bollworm, but in 2006 Bolgard 2 has been released by the Monsanto with the 2 gene double gene, that is Cry1 AC and Cry2 AB, and which protects the cotton plants against the bollworm as well as Pseudoptera caterpillar, okay. So BT cotton. So whenever we talk about the BT cotton, so it has been released in the year 1995 in the US. So how BT cotton has been developed? So the Cry gene, okay. gene synthesizing the delta endotoxin. So that has been incorporated into a cotton, and in such a way that BT cotton plant has been developed. Okay. So if you talk about the India, so BT cotton incorporated in the year 2002. So it is the first genetically modified crop which has been incorporated in India in the year 2002. So first Bolgard 1 has come, and then in 2006, in the year 2006, Bolgard 2 has come with the 2 gene, okay, for conferring the resistance against Helicobacter bollworm as well as Pseudoptera. So right now you can see, so 11.9 million hectares of area is covered by this BT cotton, okay, and there are more than 1,000 BT cotton hybrids are there, and there is a 31% of increase in the yield with 39% of reduction of pesticides, and which ultimately resulted in the increase in the profit by 88%. And right now, because of this BT cotton, okay, because of this BT cotton, India ranks first in the global cotton production. Okay, India ranks first in the global cotton production. So if you talk about the yield of cotton, so it was the stagnant for the 15 years from the year 1989 to till 2003. Okay, so before the incorporation of BT cotton. But when the BT cotton has come, so tremendously there was an increase in the yield, and we can save the crop against various insects and pests, and we can also save the cost spent on the various insecticides. So we can see here, so export and production has been increased after the incorporation of BT cotton in India, that is in the year 2002. So cotton seed oil production. So we are right now, we know that we are importing the various edible oil from other countries. Again, we are spending around 2,000 millions or 4,000 millions of Indian rupees on that, right? So cotton seed oil, it was again, it was the major concern at that time. And you know, the cotton seed oil is also, we can use it for the edible purpose. See, GM crops we cannot use, that means government is not permitting for the food crops the GM technology, but we are eating the cotton seed oil. Okay, so which is the product of that GM crop only. So this cotton seed oil production has been also increased after the 2002, that is after incorporation of BT cotton. And here we can see the edible oil consumption in India. So demand is around 24 to 25 metric ton, and we are importing around 15 to 16 metric ton of that edible oil from other countries, and our domestic production is around 9 to 10 metric ton. Okay, so whatever we are importing from other countries, whatever the edible oil we are importing from the countries, so that is of GM crops only. Okay, that is made up of soybean. So that the soybean, that means we are using the byproduct of that, but actually whatever we are importing, the base is of that GM technology or genetically engineered technology only. So soybean, you can see that GM import. So soybean, it contributes 4.90 metric ton, canola 0.50 metric ton. Okay, so what I have to say here, so we are, that means the oil which we extract from that GM crop, it's in our food chain. Okay, but we have not allowed the GM technology, genetic engineering technology, in the food crop. So so this is the data that import approval of soybean events for food and feed in India 2010 to 2014. Okay, so you can see there, so most of the soybean cultivars, they are genetically modified plants, and that has been developed for the herbicide tolerant trait or herbicide resistance trait, okay, by the company Monsanto, Bayer, BASF India. So India consumes near about 20 million tons of BT cotton seed oil. Okay, so with the incorporation of BT cotton, there is an increase in the edible cotton seed oil production since 2002, and right now we have consumed, okay, India has consumed around 20 million tons of BT cotton seed oil. That means which has been developed from that GM cotton, genetically modified cotton. But still there is an opposition to the BT cotton and various GM technology. Okay, so in beginning only I told you we have to accept the technology. Okay, we have to accept the newer technology because in green revolution we have accepted the dwarf varieties, then we have accepted the various pesticides, insecticides. Now this is the area where we have to accept such a genetic engineering technology or GM technology. So in India, efforts have been made. Efforts are also have been made to produce an herbicide tolerant cotton. Okay, so trade name was Roundup Ready Flex BT cotton. Okay, herbicide tolerant cotton, that is herbicide resistance cotton, and trade name was Roundup Ready Flex. What was trade name for that herbicide tolerant cotton? So BT plus, in addition to the BT, so one more trait will be there, that is herbicide tolerant or insecticide resistance. So the trade name was Roundup Ready Flex. Okay, so that has been developed by Monsanto. Okay, so in 2013 it was for the testing, but after that there is that technology has been withdrawn. Okay, due to some that means due to some opposition, right? But if you see right now these seeds, that herbicide tolerant cotton having a BT genes, that is coming from the illegal way for the cultivation in several states, and farmers are growing in that state. So it is happening in Andhra and various parts of Maharashtra also. So after the incorporation of this BT cotton in 2002, so there was a breakdown of that insect resistance, okay, in the year 2009. And suddenly the minor pest, that was the pink bollworm, was the minor pest in cotton. So it has become a major pest in cotton. Okay, it has become a major pest in cotton, and which has showed the resistance to both Cry1AC and Cry2AB. So it shows that this technology, all technologies have certain limitations. Okay, that means after the some interval, we have to think about the newer technology. We have to modify that technology. Okay, after a decade, we have to improve our technologies. So in India, efforts have been made to produce an BT brinjal also, okay, with Cry1AC gene. So that is for the protection against shoot and fruit borer. Okay, that is for the protection against shoot and fruit borer. So the Cry1 with Cry1AC gene, again it is from the Bacillus thuringiensis only. So the event EE1 was recommended by Genetically Engineered Appraisal Committee in 2009. Okay, so BT brinjal event means what? It is a particular, see, whenever we are producing a transgenic plant, okay, whenever we are producing a transgenic plant, means we are incorporating that transgene from a foreign source or any other species to a particular species, and that gene is getting inserted. Where that gene is getting inserted? Into a certain some part of a genome, okay, on some part of a chromosome. So that has some particular location there. Okay, so this event means what? That gene should not disturb the function of that endogenous genes. Okay, there should not be any that means there should not be deviation from the original characteristics of that cultivar where we are transforming that Cry1AC gene. Suppose XYZ variety is there, so that variety should retain its all characteristics after the incorporation of the foreign gene or Cry1AC gene. Okay, so event means, so there will be, so during the production of the transgenic plant, that we will see the various methods of transgenic development of transgenic plant in the other lecture. Okay, so that event, so it has the event name, that is EE1. Event means that particular, we can say that that particular genotype. Okay, that genotype name is what? Event EE1. We in the transgenic term we call it as an event. That means it has a particular site of insertion of that transgene on a chromosome or in a genome or in the DNA. Okay, so that EE1 event was recommended by Genetically Engineered Appraisal Committee in 2009. And this BT brinjal has been developed by Monsanto, Mexico, in collaboration with the various agriculture universities. Okay, so what was the name for that BT brinjal hybrid? So it was MHB 11BT and MHB 39BT. Okay, but unfortunately, in the year 2010, Ministry of Environment, okay, imposed 10-year moratorium for the multi-location trial of this BT brinjal. Okay, so right now 10 years are over. Right now it's there to 2022, but still now there is no decision by the ministry. Okay, so right now there is that means actually it was the Ministry of Environment who has put the moratorium on the commercial cultivation or multi-location trials of their BT brinjal. So this BT brinjal variety, so it has been developed in collaboration with the Indian Institute of Vegetable Research, Varanasi, University of Agriculture Sciences, Dharwad, Tamil Nadu Agriculture University, Tamil Nadu. Okay, so the event name EE1 developed by Mexico. So in 2010, so our Ministry of Environment put a moratorium, okay, on the multi-location trial of that genetically modified plants or genetically engineered plants. But in contrast, Bangladesh has approved that event, okay, EE1 event, same event of BT brinjal for insect resistance with Cry1 AC gene in 2030. So they have released the 4 varieties for the commercialization. But right now you can see the Bangladesh is the country where the transgenic GM food crops are farmers are cultivating these GM crops, okay, including BT brinjal. And in case of BT brinjal, there is a significant reduction in pesticide use by 28%. And farmers also getting the profit increased by 49% due to this BT brinjal, incorporation of BT brinjal. So varieties with EE1 were provided to the farmers in Bangladesh in January 2014. And actually it's coming by the illegal, that means there is an illegal cultivation in India also. That seed is coming through some illegal chains in our country. Okay. So the hybrid production in India, like BT brinjal, like herbicide Roundup Ready Flex, that is herbicide tolerant, and BT gene carrying cotton. So efforts has been made for the mustard hybrid production by using this GM technology. Okay, so mustard hybrid. Okay, so that is the creation of male steroid and restorer lines. So for development of that male steroid lines, the 2 bacterial genes have been used, that is Barnase and Bastar. Okay, so that gene has been isolated from the bacterium Bacillus amyloliquefaciens. Okay, so the efforts have been made for the hybrid development by using these genetically engineered tools, right? So various bacterial genes like Barnase and Bastar isolated from Bacillus amyloliquefaciens is used to create an used to develop an hybrids. Okay. So GM mustard. So which is the hybrid, which is right now it is ready for the commercialization. So it is a GM mustard DMH, that is Dhara mustard hybrid 11. Okay, it's almost is ready to release now, but the government is not permitting for its commercially release. Okay, so it has been developed by using the GM technology, or the male sterile or male restorer lines have been developed by using these genetic engineering tools. Okay, so there is a 30% more yield, okay, in this GM genetically modified mustard DMH 11 hybrid, that is Dhara mustard hybrid 11. Okay, so it has been developed by scientist Penton. Okay, so DMH 11. So how they have developed this GM mustard or DMH1 DMH 11 hybrid? So by using 2 genes, that is, one is a Barnase gene, okay, that has been isolated from Bacillus amyloliquefaciens. Okay, so it impedes the pollen production and making it male sterile. Okay, so Barnase gene actually produces the male sterile plant. So you know that in hybrid production we need a male sterile plant, restorer lines we need. Okay, so it hampers the pollen production and makes the plant male sterile, that Barnes gene. Okay, then this male sterile variety is crossed with the restorer plant having a bar star gene. Okay, so bar star, it blocks the action of Barnes gene. So Barnes gene is responsible for the this pollen inactivation. Okay, that means it impedes the pollen production, and that it makes the plant male sterile. But whenever we cross it with restorer lines, that restorer line is having the bar star gene, and that bar star gene again it inactivates the Barnes gene. Okay, so in such a way that Dhara mustard hybrid 11 has been developed. So there is an 27 to 30% more yield if GM mustard hybrid DMH11 will be cultivated in India. Then coming to the golden rice. Okay, again, Delhi University is involved in this production of an golden rice. So what is this golden rice? You know that rice is generally lacks its vitamin A. Okay, it's lacking the vitamin A. So Ingo Potricus is a scientist who invented this golden rice. Okay, he has used these genetically engineering tools. Then he has made a GMI, genetically modified rice, by incorporation of 3 genes. He has modified that beta carotene synthesizing pathway by incorporating the 3 genes. Okay, so which that means, and once there is an incorporation of that 3 genes in a rice, there is a synthesis of an vitamin A. Okay, there is a synthesis of vitamin A up to the level of 8 to 25 microgram per gram of rice. Okay, and general recommended dietary allowances is around 16 microgram per gram of rice. Okay, so this is the pathway where that this golden rice has been developed. Okay, so you can see there. So 3 genes, that is phytoin synthase. Okay, phytoin synthase, then phytine desaturase and lycopene beta cyclase. So 3 genes have been incorporated from the other species into the rice, and then this rice is producing the beta carotene, that is a vitamin A precursor. And now vitamin A deficient rice will produce a vitamin A. Rice will be enriched with this vitamin A content. So that is a golden rice. Okay, that is a golden rice. In general, this rice lacks these 3 enzymes. Which one? Phytine synthase, phytine desaturase, and lycopene beta cyclase. So the genes synthesizing these 3 enzymes, these 3 enzymes have been incorporated from the foreign other species. Okay, and these foreign genes have been 3 genes have been imported into this rice, which lacking this enzyme, and this rice plant now synthesizing the beta carotene, which is a precursor of vitamin A. So the golden rice, where this phytoin synthase gene has been isolated from the daffodil plant, phytine desaturase gene has been isolated from a bacteria. So phytine desaturase gene has been isolated from a bacterium, and lycopene beta cyclase gene has been isolated from daffodil plant. Okay, lycopene beta cyclase gene has been isolated from the daffodil plant, and in such a way that the golden rice plant has been developed. So India, we only talk about the BT cotton, but we should also focus the various pest resistance varieties in the pulses, vegetables. We should also focus on the herbicide tolerance in rice and wheat. We should also concentrate on the disease resistance. We should also concentrate on the nutritional quality of the various cereals. Then we should also concentrate the abiotic stress tolerance by using this GM technology in various major crops. So we should think so that the ICAR, okay, so ICAR is working under the network project that is on transgenics in various crops. So here we can see the that means insect and pest resistance transgenic crops right now are in the trials, like rice, sorghum, maize. Then the pod borer resistance trait has been incorporated in case of pigeon pea, chickpea. Then bollworm resistance trait has been incorporated in case of cotton. So similarly, delayed ripening in case of tomato, fungal resistance in case of rice and banana, virus resistance in case of various vegetables and including cotton. So the ICAR network project is working on all such traits, okay, in various crops, including edible crops. So right now we can see the various cry genes has been isolated from this Bacillus thuringiensis and incorporated into the various crops, and the various private and public sectors are involved in this. Okay, so like this insect resistance genes, various abiotic stress tolerant transgenics are being developed in the India. Okay, so in case of rice, salinity tolerance is there. Okay, for salinity tolerance, the cod A gene has been incorporated. Then for salt stress tolerance, then drought tolerance, cold tolerance, drape gene has been incorporated. Then for tomato, tolerance to salinity and drought, osmotin gene has been incorporated. So these are the various genes which are responsible for abiotic stress tolerance, have been incorporated in the various plants by using these genetically engineered tools. Then similarly, the virus resistant transgenic crops are being developed. So tomato has been developed for the various viruses like leaf curl disease virus. Okay, so by using the replicase gene, coat protein gene, antisense coat protein gene. So these are the genes: coat protein gene, replicase gene, then which imparts the resistance to the various viruses. And these plants are under the development and being developed in the laboratory level. So where we have stuck, okay, so RR flex cotton. So right now you know that Roundup Ready flex cotton is there, that is herbicide tolerance. BT brinjal, late bite resistance tomato, potato is there. Mustard hybrids, that is DMH1 is there. Protein quality potato, that is amaranthus gene has been incorporated to increase the protein quality. Golden rice has been developed to which is having the vitamin A synthesis, that means vitamin A content. Then port bedded resistance chickpea is also being ready. So but Ministry of Environment, okay, is not allowing it for the multi-location trials. Okay, so what are the major impediments? Okay, so this actually what is actually happening in case of India? So disrespecting the science-based decision, then unscientific anti-genetically modified propaganda, then fearful voice from scientific community. Okay, then the government of India halted all the field trials, including BT brinjal. Okay, so you can see now. So how this technology will come up and how you can resolve the various issues in the agriculture. So the Modi government actually they want this Niti Aayog, okay, to play a pacemaker in this GM role. Okay, so actually the KC Bansal and Deepak Pental who suppose this GM technology, okay, but Modi government, that means the government of India right now wants Niti Aayog to play a major in between role. Who are the who are opposing this GM technology and who are favoring this GM technology. So right now, so when we talk about the GM technology, okay, whenever we talk about the GM technology, so we talk about the transgene. That means some genes we are incorporating from the bacteria, some genes we are incorporating from the other plants, other species. Okay, and that genes we are incorporating into a particular host, and in such a way that we are making a transgenic plant. So in addition to the genetically modified plant, so this genetic engineering tool also used to develop a non-transgenic technology. Okay, that is by the genome editing method. Okay, genome editing. Here actually we are not inserting any foreign gene or any transgene. Okay, here we are modifying the indigenous gene of a organism. Here we are modifying only indigenous gene of a organism. So this genome editing, it is a process by which specific chromosome sequence is changed. So it involves at an insertion of at least one nucleotide, a deletion of at least one nucleotide, and or substitution of at least one nucleotide. Okay, so genome editing, actually we are not incorporating any foreign gene in the host, so that we cannot say that plant as a transgenic plant. We are only modifying the endogenous genes of that plant. So there are various genome editing technology like gene finger nucleases are there, talents are there, that is transcription activator like effector nucleases are there, CRISPR-Cas technology is there, which is widely used right now. So it is a cluster regularly interspace short palindromic repeats. Okay. So this is regarding the genome editing. So with the help of genome editing, that means the disease resistance cultivars are already in the pipeline. Insect resistance cultivars are being planted in the pipeline. Okay, virus resistance cultivars are also in the pipeline. Then various abiotic stress tolerant plants are also developed by using this genome editing technology. But right now there are some regulations. Okay, we know that in case of transgenic, so there are some regulations, like various bodies are there, like we have seen GEAC. It is an apex body which allows the multi-location trials of that GM crops. Okay, genetically engineer appraisal committee, GEAC. Okay, which allows that commercial multi-location trials of that GM crops. So there are some regulations of that commercial cultivation of that GM crops. Okay, so we have some statutory bodies like institutional biosafety committee is there. If you would like to work on recombinant DNA technology or transgenic technology, then you should first have the permission from such a your institutional biosafety committee. Okay, then there is state level biosafety committee is there. Then RCGM is there, review committee on genetic manipulations. Okay, that is then GEAC is there, that is genetically engineered appraisal committee. And finally, once the genetically engineered appraisal committee will use the permission to release a particular GM crops, then it comes to the Ministry of Environment. Okay, then it comes to the Ministry of Environment. So final decision. So there are some regulations. There are some regulations for the GM crops. That means no one can start the research for the genetically engineered plants, and that means no one can commercially release as such. Okay, there are some regulations which we have to follow, and there is some statutory bodies in India, like I said, IBC there, institutional biosafety committee, state level biosafety committee is there. Then RCGM is there, then GEAC, genetically engineered appraisal committee is there, and finally Ministry of Environment is taking the decision for this commercial release. Okay, but as I said you, the genome editing technology, where we are not incorporating a gene from foreign source, okay, where we are modifying only the indigenous genes of a plant. So is there any regulations for such a crop? Right now, can we release such a plant, or we can do India have any regulations for such a crop or for such a type of research? So answer is no. See, still now in India there is no any that means no any statutory body which can regulate these genome editing plants. So government has no any regulations. That means there is no any standard regulations to work on these genome editing plants, which we can look at at the future technologies. Okay, that means if there are some pros and cons for the transgenic plants, then we can look at the genome editing plants where we can modify the indigenous genes, where we are not incorporating any foreign gene into the plant. Okay, where we are modifying only the indigenous genes. So what should be the regulations for such a plants? So still there, the government of India is not having any regulations for that till now. Actually, draft document on genome editing organism, regulatory framework and guideline for risk assessment. So it has been proposed by the Department of Biotechnology, India, to the Ministry of Science and Technology. Okay, in January 2020, but till now there is no any decision from the government. Okay, for such a how to work on such a genome edited organism where we should consider them as a transgenic or where we should not consider them as a non-transgenic, or shall we follow the same procedure whatever we are following for the release of transgenic plant or working on the transgenic plant like IBSC, GEAC, RCGM, state level biosafety committee. So there is no such a any there are no such a any regulations and guidelines from the government to work on such a genome edited crops. So with this I can with this I can stop here. This is regarding the status of genetic engineered crops, okay, across the nation, and what's happening in India, what's happening at national level. So you should look at this. So this genome editing, actually, if there is some problem with for the release of transgenic plants, so government should look at this non-transgenic technology like genome editing technology. Okay. So if you would like to discuss with me anything regarding whatever I told you, whatever the traits I have explained you, and the various cultivars, the trade name which has been used for the GM crops, you can discuss, or you can ask me any questions if you have. So you can unmute yourself and you can ask the question, or you can type it in chat box.
This topic is open for discussion. If you have any queries regarding this topic of curiosity, so please discuss with Dr. Yogesh sir.
If you have any queries, you can ask it. So you should know about the first GM plant, first GM plant in cereal, first GM plant in forestries, first GM plant in the case of flower, then which is first commercially released GM plant, which is the country which is highest cultivating this GM plants, which is the major trait, that is herbicide tolerant trait is that means if you consider worldwide, then it comes first, the herbicide tolerant trait. Then which country is growing the largest that means in which country there is a largest cultivation of this GM crop. So you should remember all such things. So when the BT cotton has been come in India, then when Bolgar 2 has come, so which are the genes are there, from where they are isolated, what is the status of BT brinjal, then in BT brinjal which event has been approved, then GM mustard. So the GM mustard is also ready for the release. So that is the that means hybrid has been developed by using genetically engineered tool, that is a DMH111 hybrid, that is Dhara mustard hybrid. Then golden rice, what actually it is golden rice, so it is enriched with this vitamin A. So generally rice lacks the vitamin A, and there are 3 genes which has been incorporated from the foreign source. You can type it in chat box also, you can, if you have any doubt. Yeah, sure, I will send you the notes also, and I will also take the quiz also. I will give the some MCQs to the Bhalerao sir to have one quiz on this genetically modified plant. Okay, I will provide this with answer also. You can also see the answer after some time. So that multiple choice questions I will provide you, so that that means you can practice it. Okay. So you should remember that various statutory bodies are there in India, so which regulates the genetic engineering, that means genetic engineering research in our country. Actually, we are consuming lot of GM products. Okay, even though our government is not permitting the GM plants for GM food crops for cultivation, but indirectly we're eating it at larger level. Okay, so you should remember everything.
So there is one question in chat box, sir.
Yes.
What is the difference between marker used crop and genetically modified crops?
See, genetically modified crops, what I told you, the genetically modified crops are those where we are incorporating the gene from the other source or other species. Okay. So that is a transgene. Okay. We call it as a transgene. Okay. So in molecular marker developed plants, we are not incorporating any foreign gene there. Okay. We are not incorporating any gene from the other species. In case of these two technology are there by using molecular. These all are genetic engineering tools. By using molecular markers, we can do the crop improvement. By using transgenic approach, we can do the crop improvement. Okay, there are two ways of that crop improvement. First is by transgenic, where by using transgenic we can take a gene from other organisms and that we can import it into a crop plant for various traits like her insect resistance, herbicide resistance. By using molecular markers, okay, by using molecular markers, you can have the gene from the same species. Okay, we are only tagging a gene, we are using it for the breeding purpose. Okay, so molecular marker right now, improved Pusa Basmati is the variety which has been developed by this molecular marker technology, which is resistant to the bacterial blight, which is resistant to the blast disease. It has been developed by our Honorable Director General of ICAR, Dr. Trilochan Mahapatra, and it is in commercial use. The variety name is improved Pusa Basmati. Then,
sir, kindly repeat, sir. Voice was breaking. I was not able to hear.
so improved Pusa Basmati is the variety. Right now, can you hear right now?
Yes, sir. Yes, sir.
So
Thank you,
improved Pusa Basmati, okay, Suvarna, they are the varieties of rice which has been developed by using this molecular marker technology, where we are not incorporating any foreign gene into an plant. Okay, we are only, this is the tool, molecular markers are the tool which hasten the breeding program, okay, which minimize the, that means the breeding program, that means we tag them in breeding program, we do the crossing, then F1, F2, F3, F9, it takes several generations, we have to wait for 9 to 10 years. But with the help of molecular marker technology, we can have the that means we can finish the breeding program within 2 to 3 years. Okay, but we are not incorporating any foreign gene. Okay, here we are using the wild relatives. Wild relatives means we can, that wild relatives, some plants are having insect resistant gene, some plants are having disease resistant gene. So with the help of molecular markers, we can incorporate that gene into a cultivated crop. Okay, cultivable crops. So molecular marker technology, where we are not incorporating any foreign gene or any other gene from the other species. But in case of transgenic, we can import gene from bacteria. You can take the gene from other flower crop. Like in case of golden rice, we have seen that gene has been incorporated from the daffodil, which that means that for vitamin A synthesis in golden rice. That means we can. This is the other species where we can import the gene. And in case of molecular marker technology, where we take the gene from the same species of and that wild plant, and we hasten that breeding program. So it is related only the breeding program. So the improved Pusa Basmati and Suvarna, these two varieties are there, which has been right now they are commercialized. Okay, they are the product of molecular marker technology. Improved Pusa Basmati have the 3 genes for bacterial blight resistance, bacterial blast disease resistance. Okay, there are 3 genes. They have been incorporated by using these genetic engineering tools. That is a molecular marker technology. Suvarna variety is there. Suvarna variety is Suvarna variety of rice. It has a character of submergence tolerance. The sub-1 locus has been incorporated from wild relatives to the cultivated crop. Okay, so this is the difference between molecular marker technology and transgenic technology.
Sir, there is another question in chat box. Sir,
Yes.
any chance of gene escape from the genetically modified crops?
See, gene escape. Okay, so that there are horizontal gene flow is there. Then so horizontal gene flow means that that some genes may get transferred from their close relatives. Okay, So there is a chance, okay, there is a chance. But whenever we release an GM crops, whenever we ask the farmers to grow particular GM crops, okay, so as every technology has its pros and cons, okay, so we are giving some guidelines how to grow these GM crops. We have to use the refugee. So what should be the isolation distance? So everything has been, if you follow the proper recommendation, then there is no chance of gene escape. If you follow proper recommendation, then there is no chance of a gene escape. If you will not follow recommendations, then all technologies will get failed, including GM crops, even any technology. If you will not spray the pesticide at optimum level, so what is the use of that pesticide then? So it's like that. If you should follow the whatever the recommendations given along with this technology, okay, then only it is beneficial.
So there is another question.
Yes.
Sir, what is the difference between marker and primers?
See, markers and primers, okay. So markers means what? So the primers, okay, first I will talk about the primers. So the primers are nothing but they are the short oligonucleotide. This is a short nucleotide sequence. You know, the genes are made up of nucleotides, okay, or DNA is made up of nucleotides. So that gene, okay, if you would like to produce multiple copies of that gene or that particular segment of a DNA, okay, we use the markers, okay, that is the sequence which is complementary to that target strand, okay, which is complementary sequence to that DNA, and which we are using to produce that particular segment, DNA fragment or DNA segment or a gene, to produce in multiple copies of that. And markers, when we talk about the markers, these are the primers only, but that primers are tagged with the particular trait, okay. They are identified and identified for a particular location in a gene. They are identified for a particular location on a chromosome. They are identified for a they are identified with linkage with particular trait like yield, like disease resistance, like insect resistance. Whatever I have given the example of that improved Pusa Basmati. Okay, so markers are nothing but the primers only. But the markers, when we call them as a markers, when they are there, we know their exact location in a genome or on a DNA or on a chromosome, and these markers are linked when we identified them for their linkage with a particular trait. It may be either insect resistance, disease resistance, or any yield or quality trait. Okay, so they are nothing but the primers only. We are using this primer as a markers. Okay, we are using that primers only as a markers for the breeding purpose, and that is by the molecular marker technology.
Okay. So next question is, sir, why India not allowing genetically modified crop?
See,
Genetically modified food crops for commercial cultivation.
See, actually, this answer only the government can give, but I can tell you because see, what examples I have given to you, the edible oil, okay, cotton seed, that is cotton seed oil we can consume, okay. We are consuming we are importing the edible oil from foreign countries. They are that whatever the oil, that is soybean oil, which contributes 50% of that, and that soybean is only the GM soybean. But ultimately it's in our food chain, okay, it's already in our food chain. I have given the good example of Bangladesh. They are cultivating the BT brinjal since 2006, but still there is no issue. The farmers are benefited 40% more. And in India, if we talk about the India, there is a regulatory mechanism, that's fine, okay, but actually there are that means a lot of controversies are there. That means there is one group which oppose these transgenic plants, and there is one group which favors this transgenic technology. So but actually it's the decision which is at the level of government, okay. If they are not permitting the GM crops, GM food crops, so they at least they should give the permission for genome edited plants. That is a non-transgenic technology, okay, that is non-transgenic technology. At least they should give the permission for such a genome edited plant where we are not incorporating any transgenic plants. But at least they should finalize such a regulations. They should finalize such a rules and regulations for the genome edited plants. Like in transgenic plants, there are regulations, but at least there should be some regulations whether that genome edited plant should be considered as like transgenic, or they should follow the same procedure as like transgenic through various statutory bodies like GEAC, RCGM, Institutional Biosafety Committee, like that.
Okay, sir. So is there any question? I think this is a fruitful outcome, sir. This is a very nice and wonderful lecture you delivered, sir.
Yes.
And most of the participants interact with you in this regard. So I think this is a burning issue, genetically modified crops, and most of the techniques we are using in this technology. So this is big. And which type of the question will arise on the NET or ARS examination? So next lecture, when you will take the next lecture, sir. So at that time you can cover that type.
Yes, sir. I'm covering the various methods for used for the genetically modified plants.
Yes, sir. Yes, sir.
And I will also provide
And,
the question. That means we will have this one quiz in the Google form.
Yes,
so that I will provide that link also. Okay.
Yes, sir.
Yes. So that
Most of
also,
the people demand give some reference. So which book is useful for genetically modified crops? So they may also ask.
Right now, if you think about the competitive examination, so a textbook of biotechnology by Jain, okay, JAIN, okay.
Jain.
Jain. So that book you can refer. And if you consider the if you have to see with respect to the competitive examination and concept, if you have to clear your concept, then BD Singh is okay.
Yes.
A textbook of biotechnology by BD Singh.
Yes, sir.
But actually you should and see for status, if you would like to see the status regarding the GM crops and all updates of GM crops till now, till today, so you can visit the IES AAA website. Okay, IES AAA. So there is a, can you see my screen? Can you screen?
Yes,
screen?
Yes, sir.
Yes, your screen is visible, sir. Nice.
See, International Service for Acquisition for Agribiotech Application. So if you go here, so all updates you can see here. So what is the area of biotech crops? Which trait is see here, GM approval database also you can see in which country which crops has been developed. Okay, for which traits has been developed. You have to select crop wise, you can select country wise, you can select trait wise. Suppose if I select the crop, suppose if it is a cotton, so it is a commercial trait where you have to follow, see the for the herbicide tolerance, insect tolerance, or other trait. So if I visit her, that you can see, sir, there.
Yes. Sir.
So this IAS AAA website, you can see all update you will get here. So everything whatever updated in the field of genetic engineering, so you will get it.
Everybody got this website address, sir, and they will search some information on that particular website, and it will be very helpful in future also. So
See,
thank you very much, sir,
okay,
for such a wonderful lecture, and accepted our invitation. On behalf of the Department of Extension Education, Postgraduate Institute, Mahatma Phule Krishi Vidyapith, Rahuri. So we are very much thankful to you, sir, for delivering such a nice lectures. And next lecture we will also arrange.
Okay, sir. Okay.
So, thank you very much, sir.
Thank you, sir. Okay.
Okay, sir. Thank you, sir.
Yes.